welcome to Biosumit. Today we learn about CRISPR Cas9 system. So we know that it's a site-specific gene editing technology, and nowadays it is widely spoken and widely remembered because recently two scientists received a Nobel Prize on CRISPR Cas9 work. So today we learn about what is the basics of CRISPR Cas9. So this is an editing technology of gene, okay, but actually this is a system of this is a part of adaptive immunity of prokaryotic system. CRISPR means uh, clustered regularly interspaced short palindromic repeats. Palindromic repeats means suppose you are reading one DNA frame in from 5 prime to 3 prime direction, and the same if you are reading from its complementary strand from 5 prime to 3 prime, the frame will be same, okay, like this same base sequence. Like in terms if I say like the word is 10-8, so if you are reading from left to right or right to left, it will be same. So this is called palindrome. In case of DNA, it will be from the both strand from the 5 prime to 3 prime direction, you can read the same, okay. So this is called palindrome. Now first, it is reported in both of the bacteria in gram positive and gram negative. Now with CRISPR there is another thing, that is Cas9. So what is Cas9? This is an endonuclease, and endonuclease is what? It breaks the DNA. This Cas9 breaks actually double stranded DNA. So this is the this is Cas9, okay. Now let's check this diagram. Here you can see I have drawn some boxes. Let's say this is the palindromic repeats, okay, and I am showing here this is the bacterial genome, or you can say plasmid, okay, and in between you can see there are some spacer, okay, this is red, this is black, these are spacers. So the palindromic repeats are the indigenous properties of the bacterial genome, okay, and these spacer, these are foreign. Actually these are gained by viral infection, or can be transposon, or any type of virus like bacteriophage, etc. So this is not the indigenous part of bacterial plasmid, okay. This is one kind of memory, you can say, like what happens in human also. You can you know that in case of vaccination, there are some viruses are previously from beginning, before the infection you are introduced with some viruses or some, you can say, pathogens, and your immune system creates a memory of that infection, and later if you are getting that infection, so this adaptive immunity will help you to help you from prevention of that disease. So this is a type of memory actually. So bacteria is taking the viral infection previous memory and inserting it to its plasmid. This is called spacer, okay. This is, remember, this is the foreign DNA, this is the recognition of that previous attack to defense in future. So what happens when a virus attacks a bacterium? So let's say there is a phage virus is attacking this bacteria, and this is its viral DNA or viral genome, okay. So what will happen here? This part will be inserted to the bacterial plasmid, okay. This is the, as I have told you, spacer. Here you can see this is also a spacer, this black colored portion. So it can be said that there may be two viral infections, and these are two viral infection memories, and these are the repeats. So as I have said, in between repeats the spacer will be adjusted, okay. The blue one is repeats, and we call it CRISPR array, okay. Now what will happen? This is a plasmid, so it will go for transcription, right? So now we will get these are the repeats, these are spacers, and this is also spacer. So this is called precursor CRISPR RNA, or in short pre-CR RNA. Okay, now here will be added tracer RNA. Tracer RNA is actually the complementary to the repeats. Here you can see the blue ones are the palindromic repeats, and these parts, the complementary parts of these repeats will be like this, that will be tracer RNA. And with the help of Cas9, there will be some complex formed. Let's say this is the tracer RNA. Okay, now the tracer RNA will bind like this, and here is the here is the spacer. So this is tracer is binding to the palindromic repeat, this is the spacer, and there are some other you can say there's some other tracer RNA with is binding to the palindromic repeats with having other spacer. Okay, and it will be surrounded by Cas9. Okay, and like this. So here you can see, if I am considering it as a complex, so here you can see palindromic repeats with the complementary part of H, that is called tracer RNA, with the spacer sequence, that is the foreign part, that can be from viral infection, and surrounded by Cas9, that is a nucleus, and here is also the same. So now with the help of RNase 3, this part will be chopped. Okay, so here you can see this will be an independent complex from the whole strand, right? And this will be some independent complex. So these independent complexes are called effector complex. So effector complex is composed of free CR RNA, sorry, I am writing it here, pre-CR RNA, tracer RNA, and Cas9. These three will form one effector complex. So there will be so many effector complex, and that can be the memory to defend itself. I mean, the bacteria will defend itself from further infection of any virus or something like that. Okay, so let's say that virus, this virus is affecting again this bacteria with, remember, these bacteria now have this effector complex, right? So let's say this is the cell, and now there is phage, and this is phage virus. Okay, now what happens, the bacteriophage will replicate its phage to the host, right? So let's say we are making it bigger in here, the DNA replication will happen like this, right? The two strand like this. Okay, this is the first DNA. Okay, this is replicating in the host, that is in bacteria. Now the effector complex, remember the effector complex has this sequence, right? This is called spacer, so it knows the same sequence, so it can recognize this sequence, and it must be the some part will be the complementary to its new and to the new virus, to the new genome of this virus, right? So previously these are the spacer. So this effector complex will recognize the part. Now this is the previous repeating part of the bacteria and the tracer RNA part. Remember the tracer RNA part with the what? With spacer, right? So this spacer will recognize the part of any genome of that newly affecting virus, correct? So this will form a complementary sequence here, and this is surrounded by a Cas9. Okay, this is Cas9. Now here a short unique sequence that is unique will be to this viral genome will be recognized here. Okay, suppose this. Okay, this is called PAM or protospacer adjacent motif. Okay, so what is happening? The same phage is attacking this bacteria, and it's now ready to replicate, but the complementary part of the spacer is binding to its complementary part, and with this effector complex here PAM is recognized. And what happens after recognizing PAM to its upstream? What happens in the both strand to its upstream? The snipping will occur. So after recognizing, what happens after recognizing the PAM in the upstream of this PAM, in the both strands the snipping will occur by the endonuclease. So what happens, the virus will be get neutralized and it can't do its replication, and that's why it will not be able to affect the bacteria, and that's why the bacteria will be safe from its infection. Okay, so recognizing its PAM. So this is how bacteria protects itself from viral infection, so creating the memory. So this is the effector complex, again I am telling, and the part of the spacer, the spacer part of the effector complex will make the complementary to its affecting virus, and then the PAM will be recognized, and in the upstream of this there are some sequence in the both strand that will be snipped, and the virus will be neutralized, and it can't affect, as its genome can't do the transcription and can't make some other RNAs. That's why it will be neutralized and the bacteria will be safe from its effect, right? So this is the general part of the CRISPR, how it's work in bacteria. Now how it is used or utilized in the genome editing technology of eukaryote and any other eukaryote systems, that will talk in the next video. So how it's happening, like non-homologous end joining or some other methods, so we'll talk about in the next video. So till then, bye, thank you.